This is Chapter 12, Module 2, Neurons and Neuroglia. The learning objectives of this module are 1. Describe the structure of a typical neuron and discuss the function of each component. 2. Classify neurons based on their structure and function. 3. Describe the locations and functions of the various types of neuroglia. And 4. Explain how the brain and the spinal cord are protected.
Neurons are nerve cells that are specialized for intercellular communication. Neurons have a variety of shapes. A multipolar neuron is the most common type of neuron found in the central nervous system. Each multipolar neuron has a large cell body, several short branch dendrites, and a single long axon ending in terminal branches called telodendria. The cell body, also called soma, contains a large round nucleus with a prominent nucleolus. The cytoplasm surrounding the nucleus is the perikaryon. The cytoskeleton of the perikaryon contains structures similar to the filaments and microtubules of other types of cells. The perikaryon also contains mitochondria, used to make ATP to meet the high demands of the active neuron. Ribosomes and rough endoplasmic reticulum are also found in the perikaryon, and they help produce proteins. The rough endoplasmic reticulum and the ribosomes cluster together, and these regions are called Nissl bodies, which give the neuron cell bodies a gray color, referring to the gray matter that is seen in dissection of the brain and spinal cord. Most cells don't have centrioles, the organelles needed for the formation of the spindle apparatus in mitosis. Therefore, neurons can't divide. Therefore, if a neuron is lost to injury or disease, it can't be replaced. The slender extensions off the cell body are called dendrites. Dendrites receive information from other neurons. The axon is a long process off the cell body that is capable of propagating an electrical impulse known as an action potential. The cytoplasm of the axon is called the axoplasm, and the plasma membrane of the axon is called the axial lemma. The axial lemma is a specialized portion of the plasma membrane. In a multipolar neuron, the axon has a thickened region close to the cell body called the axon hillock. The axon hillock leads to the base of the axon, or the initial segment. An axon may branch along its length, producing side branches called collaterals. Collaterals enable a single neuron to communicate with several cells. The main axon and the collaterals may end in a series of fine extensions called telodendria, or terminal branches. These telodendria end at the synaptic terminals, also called synaptic knobs. The synaptic terminal is part of a synapse, which is a specialized site where the neuron communicates with another cell. Each synapse involves two cells: the presynaptic cell, which is the cell that sends a message and includes its synaptic terminal, and the postsynaptic cell, which is the cell that receives the message. The space in between the synaptic terminal of the presynaptic cell and the postsynaptic cell is called the synaptic cleft. Chemicals contained in vesicles in the synaptic terminals will be released via exocytosis into the synaptic cleft. This release is triggered by an action potential traveling down the axon of the presynaptic cell. The postsynaptic cell can be another neuron, or it can be another type of cell. One neuron may communicate with another at a synapse on a dendrite, on the cell body, or along the length of the axon of the receiving cell. A synapse between a neuron and a muscle cell is called a neuromuscular junction, as we saw when studying muscle tissue. Neurons may also innervate, or are distributed to, a variety of other cell types such as glands and adipocytes. The postsynaptic cell contains receptors for the neurotransmitters. The neurotransmitters bind to these receptors. The synaptic terminal at a neuromuscular junction is more complex than between two neurons. Neuroglia are the cells in neural tissue that support and protect neurons. Neuroglia are abundant and diverse, and they account for roughly half of the volume of the nervous system. The central nervous system contains a greater variety of neuroglial cell types than the peripheral nervous system. We have only a limited understanding of the functions of these cells. The central nervous system has 4 types of neuroglia: ependymal cells, astrocytes, oligodendrocytes, and microglia. The peripheral nervous system has 2 types of neuroglia: satellite cells and Schwann cells. Ependymal cells line the central canal of the spinal cord and the ventricles of the brain. They assist in producing, circulating, and monitoring cerebral spinal fluid. The central canal of the spinal cord is a fluid-filled passageway that extends along the longitudinal axis of the spinal cord in the brain. Cerebral spinal fluid fills this passageway and also surrounds the brain and spinal cord. Cerebral spinal fluid circulates continuously and provides a protective cushion to the brain and spinal cord, as well as circulates dissolved gases like oxygen, nutrients, wastes, and other materials. The ventricles of the brain are also lined by ependymal cells. The ventricles are enlarged chambers found in several regions of the brain. The ependymal cells that line the central canal of the spinal cord and the ventricles of the brain form an epithelium called ependyma. Astrocytes are the largest and most numerous neuroglia in the central nervous system. These cells have a variety of functions. Many of them are poorly understood. They help to maintain the blood-brain barrier, provide structural support, regulate ion, nutrient, and dissolved gas concentrations, absorb and recycle neurotransmitters, and form scar tissue after injury. Astrocytes maintain the blood-brain barrier. Compounds in the blood do not have free access to the central nervous system. The cells lining the central nervous system capillaries create a blood-brain barrier, and they control the chemical exchange between the blood and the interstitial fluid. These cells essentially isolate the central nervous system from the general blood circulation of the body. The astrocytes have slender extensions that wrap around capillaries. These processes form a complete blanket around the capillaries except areas where the neuroglia come in contact with the capillary walls. Oligodendrocytes have slender cytoplasmic extensions, but the cell bodies of oligodendrocytes are smaller, with few processes than astrocytes. The processes of oligodendrocytes are in contact with the exposed surface of neurons. These processes completely surround the axons in the central nervous system and insulate them from contact with the extracellular fluid. The tip of each process expands to form an enormous pad which resembles a pancake. This flattened pancake gets wrapped around the axon, forming concentric layers. This wrapping is called myelin. The myelin wrapping serves as an electrical insulation and increases the speed of the action potential traveling along the axon. When an axon is wrapped in a myelin sheath, this axon is said to be myelinated. Each oligodendrocyte myelinates segments of several axons. The large areas of the axon that are wrapped in myelin are called internodes. The gaps in between the adjacent internodes are called nodes or nodes of Ranvier. In dissection, myelinated axons are glossy white because of the lipids in the myelin. As a result, regions in the central nervous system that are dominated by myelinated axons are known as white matter. Not all axons of the central nervous system are myelinated. Unmyelinated axons are axons that are not completely covered by myelin. Areas of the central nervous system that contain these unmyelinated axons, as well as the cell bodies of the neurons and their dendrites, both of which are unmyelinated, have a gray color and make up the gray matter of the central nervous system. Microglia are the least numerous and the smallest neuroglia in the central nervous system. These cells are phagocytic. They can migrate through the neural tissue. They form during embryonic development from stem cells that also produce monocytes and macrophages. They migrate into the central nervous system during embryonic development as the nervous system forms. They remain in the central nervous system throughout life, acting as a wandering janitorial service and police force by engulfing cellular debris, waste products, and pathogens. The peripheral nervous system also has neuroglia. There are two types of neuroglia in the peripheral nervous system: satellite cells and Schwann cells. Satellite cells surround neuron cell bodies in the ganglia. They regulate the environment around the neurons, much like the astrocytes do. Schwann cells form a sheath around peripheral nervous system. A Schwann cell can myelinate axons, but only a segment of a single axon. In the central nervous system, oligodendrocytes were able to myelinate portions of several adjacent axons. A series of Schwann cells is required to enclose an axon along its entire length. In the peripheral nervous system, Schwann cells play a part in repairing damaged nerves. In a damaged nerve, the axon distal to the injury site degenerates in a process known as Wallerian degeneration. The Schwann cells do not degenerate, and they proliferate to form a solid cellular cord that follow the path of the original axon. As the axon grows alongside the appropriate cord of Schwann cells, it may eventually reestablish its normal synaptic contacts. If it stops growing or wanders off in a different direction, normal function will not return. Limited regeneration can occur in the central nervous system, but the process is much more complicated because there are many more neurons involved, and the astrocytes may produce a scar tissue that prevent axon regrowth across the damaged area. Astrocytes may also release chemicals that block the regrowth of axons.
This ends Chapter 12, Module 2, Neurons and Neuroglia.